Figure 2.11 describes the relationship between photosynthesis and light, or as it's better called, irradiance. It's describing a photosynthesis irradiance curve, or as it's more often referred to, a PI curve. It's important to remember that although photosynthesis is light dependent, respiration is more or less constant. With increasing light, photosynthesis increases linearly with a slope of alpha, and a term for photosynthetic efficiency, until it reaches a particular irradiance where the photosynthetic rate is equal to the respiration rate. This point is called the compensation irradiance, EC on the graph, or the compensation point. The irradiance where this occurs is very species specific, and even within a single species can vary with season or even on shorter timescales of months or days. As irradiance increases further, the trend becomes gradually non-linear, and a point is reached where further increases in irradiance do not result in increases in the photosynthetic rates. In other words, the rate of photosynthesis is light saturated, and the maximum rate of photosynthesis, Pmax on the graph, has been reached. The saturation irradiance, EK, is the irradiance at the intercept of the two lines shown. In some organisms, there's a decrease in photosynthetic rates at very high irradiances, so-called photoinhibition. This is a result of damage to cell membranes or proteins by the high light. It doesn't happen in all species and generally is in very high irradiances indeed. Two other important terms need to be noted: gross photosynthesis in the graph PG, which is equivalent to the total photosynthesis, and net photosynthesis PN, which is equal to the gross photosynthesis minus the respiration. The distinction between these two is very important, since it's only really the net photosynthetic energy gains that are available for cell growth and reproduction. In panel B, there's some experimental PI curves are shown. The red data are from cells grown in the shade, which induces higher concentrations of chlorophyll A per cell. The blue data are from cells of the same species grown at high light with relatively less chlorophyll A per cell than the shade acclimated cells. Looking at the top panel and comparing the PI curves of the shade and light acclimating cells, it would indicate that the Pmaxes are very different. The Pmax of the shade cells being very much lower than the light acclimated cells. However, looking at the bottom panel, where the rates of photosynthesis are expressed on a per cell basis, so the chlorophyll differences have been removed, the Pmaxes are actually identical, and it is the alpha values, or the efficiency of photosynthesis, that are different. This example really highlights the need for units to be chosen carefully for interpreting the data.